Hello everyone, welcome to Chem 9 lab session. This week will be our final lab experiment and we will learn about amino acids. The experimental instructions are given in this PDF file and the sample data needed for the lab report is provided in this section. So let's take a look at the experimental instructions first. This lab experiment has two sections. In part 1, we will learn about the structure of amino acids, and in part 2, we will learn about a technique called thin layer chromatography. And using this technique, that is thin layer chromatography, we will determine the identity of an unknown amino acid from the list of amino acids whose data is provided to us. So amino acids are the building block of proteins. In human body, we need about 20 different amino acids to function properly. However, the body cannot synthesize all 20. So those amino acids that need to be ingested are called essential amino acids. Now these compounds are called amino acids because they contain an amine group, which is the NH2 group. As we can see, this group is the amine group, and they also contain carboxylic acid group. Hence these compounds are known as amino acids. So both the amine group and carboxylic acid groups are connected to a carbon atom, and this carbon atom is known as alpha carbon atom. The alpha carbon atom will be linked to a hydrogen atom and a group. In this general structure, this group is labeled as R. In all 20 essential amino acids, the R group is different. The rest of the amino acid structure remains the same. That is, this part is same in all amino acids. The R group is different. A list of common amino acids are given here, and as you can see, in case of glycine, the R group is just a hydrogen. In case of alanine, the R group is methyl, and in case of phenylalanine, the R group is a benzene ring with CH2 group. Similarly, you can see different amino acids have different R group, but the remaining part remains the same. Let's go back to the general structure of amino acids. In organic chemistry, we learned that the amine group is a base or a basic group, and the acid group or carboxylic acid or carboxyl group is an acid group. As such, in this form, amino acids are neither base nor acid because base and acid groups kind of neutralize each other out. We also know that the bases can accept a proton. That is the Bronsted-Lowry definition. Proton is the H positive ion, and acids, they donate a proton. The form which is shown here, so this is the neutral form. Now, because the base group can accept a proton and acid can donate a proton, therefore amino acids undergo a proton donation process, which is shown here. So in this proton donation process, the proton from carboxylic acid is donated to the amine. So in other words, the carboxyl group will lose a proton as you can see, there is no proton here, and the amine group will gain a proton. So we can say that a proton is transferred from the carboxyl group to amine. So two things happen when this transfer takes place. So first thing is that the carboxyl group becomes negative charge and the amine group becomes positively charged. So because of these two ions present, this kind of the structure is known as Zwitter ion. Zwitter ion is an ion which contains both positive and negative within the same molecule. And it's a dipolar ion because it contains positive and negative, so therefore there will be a dipole. Now because both positive and negative charges are present within the same molecule, so it is actually a neutral molecule because positive and negative cancel it themselves out, but it contains a polar. So this kind of structure of any amino acid is known as Zwitter ionic structure. So that's what happens when a proton is donated from the carboxyl group to amine group, and it happens to all the amino acids. The another thing to consider is what happens when these amino acids are in acidic solution and what happens when they are in a basic solution. So in acidic solution, when the pH is very low, we know that in acidic solution we have excess of protons. So then what happens is that in acidic solution, the carboxylate ion this part, the COO part, will gain a proton and it will become COOH negative in acid solution. So in acidic solution, the COO negative, the negative charge will disappear and it will become COOH, but the amine positive charge remains the same. So that's what it says. The carboxyl part becomes COOH in acidic solution, but the ammonia or amine part retains its positive charge. So in acid solution, the amino acids will be NH3 positive, but the carboxylate group will become COOH. So it loses its negative charge. Opposite thing will happen in basic solution. In basic solution, the COO negative will remain COO negative and the amine group will lose its H positive and it will become NH2. So this happens in a basic solution. In neutral solution, both COO negative and NH positive remains with their respective charges. So this is in neutral solution. Okay. So this is important because in the lab report we need to write the structures of these amino acids in their neutral zwitterionic state. So this one. Then we have to write the structure of amino acids when they are in acidic solution. Remember, in acidic solution, the COO negative will change to COOH and NH3 positive remains the NH3 positive. And then we also need to write the structure in basic solution. And remember, in basic solution, the COO negative part remains. However, the positive ammonium part will change to NH2 neutral. It will lose its positive charge. So this will be the first section of our lab report in which we note down or we write down the structures of various amino acids, and the structures of these amino acids are given in this table. So you will take a look at this table and find the structure. All these structures are for in zwitterionic or the neutral state. You can see that they have both CO negative and NH3 positive parts in all of these. And in acidic solution, you have to write down the structure of these required amino acids in acidic solution, and then also in basic solution. Some other properties of amino acids are also given in this table, where the important information about this amino acid is the symbol. For example, glycine, the symbol is either GLY or just G, and also the polarity of these amino acids. Some of the amino acids are non-polar and some of them are polar. So all the information needed for the first part of lab report is provided in this table. The second part of this lab report deals with the technique called TLC, or thin layer chromatography. A video about thin layer chromatography is given here, so I would highly encourage you to watch this video. So let's briefly discuss what are we going to achieve in the second part of this lab report. The TLC technique is based on the fact that various compounds have different amount of polarity, and depending on the polarity, solubility of these compounds within a solvent is slightly different. So we can apply this concept to identify the identity of unknown compound as shown here, provided we have a data available from various samples. So in the TLC experiment of amino acids, we have 6 different amino acid solutions available to us. And these 6 different amino acid solutions are labeled as number 1, 2, 3, 4, 5, and 6. Identity of these 6 different amino acids are given in the sample data. For example, number 1 is phenylalanine, number 2 is alanine, number 3 is glycine, number 4 is serine, number 5 is lysine, number 6 is aspartic acid, and then we have an unknown amino acid. The key point here is that unknown amino acid is one of these 6. We don't know which one, but it is one of these 6. So using TLC, we can find what this unknown amino acid is. So what we are going to do is we are going to deposit our 6 different amino acid samples on this piece of paper. So this square represents a piece of paper. It's like a filter paper. The dots represent a droplet of our amino acid solution. So when you put a droplet of amino acid solution, the solvent will evaporate eventually, and the amino acid will stay on the filter paper, and we call it that the sample has been deposited. So we will deposit our 6 amino acids and the unknown. So 6 amino acids are deposited here and the unknown is deposited here. And remember, this square represents a filter paper. Then we will fold this filter paper like this, and then we insert or put this filter paper in a chamber which contains a solvent. Now the level of solvent has to be below this spotted line. The idea is that the filter paper will absorb this solvent, and this solvent will start rising up on the filter paper. As the solvent is moving up, it will start dissolving the amino acids, those spots, and those amino acid spots will also start traveling. Once the filter paper reaches almost to the top, we will pull this filter paper out. And when we pull this filter paper out, then we let it dry. And we will get a sample something like that. So this is a small cross section of that filter paper. It's not showing all the 7 spots, it's only showing 3 spots. The rest of the filter paper will behave the similar way. So what happens is that the solvent started from all the way from here and it reached to the top. So this is the distance traveled by the solvent. As solvent was moving up, the spots which were initially down here, those spots also traveled. For example, spot A traveled from the bottom all the way up here. So this is the distance traveled by sample number A or sample 1. So from these two distances, we calculate a value or a parameter called RF parameter. So RF parameter is given by the distance traveled by the sample, which is spot. In this case, if it's A, so for example, A traveled 4.1 centimeter and the solvent distance was 6.8. So the RF value of A is 4.1 divided by 6.8 or 0.6. So that's how we calculate the RF value. So applying the same logic, we will calculate the RF value for all of these 6 amino acid spots. They will be all the way up or down somewhere. And how much amino acid spot gonna travel on the paper, it depends on its polarity or the solubility in the solvent. So no two compounds have exactly the same RF value. So maybe number 1 will travel up here, maybe number 2 will here, 3 will here, 4 will here. So we will calculate the RF value, and we will calculate the RF value of unknown also. The RF value of unknown will match with the RF value of one of these 6 samples, and whichever RF value it matches up with, that is the identity of that unknown sample. So that's the entire experiment, and that's how we calculate or we figure out the identity of unknown amino acid. Let's take a look at the lab report. As usual, the sample data is provided in this tab, and the lab report is provided in the final section. Let's open the lab report. So in the first section of the lab report, we have to draw the 3 condensed structures of glycine in following conditions. So first condition is ionized form, and as we saw in the PDF file, the ionized form data is given, and then we have to write the structure in acidic solution and then in basic solution. We already learned how to write the structures in acidic and basic solutions. So draw these structures, draw these formulas on a piece of paper, and then upload the picture of that paper here. And then you have to find if glycine is polar and non-polar. Again, this information is given in the PDF file in that table, and whatever you find, just select that. After glycine, you have to do the same thing for alanine and then for serine. So these 3 amino acid structures has to be drawn in ionized form, acidic solution form, as well as in basic solution. And then you have to find out whether they are polar or non-polar. And this should be the first section of your lab report. Second part is the chromatography of amino acids, or the TLC, or thin layer chromatography. In this section, first you have to draw the chromatogram and indicate the color of each spot, and then you have to upload the image. So how are we going to draw the chromatogram? We have to draw the chromatogram based on the sample data. Let's take a look at the sample data. So when you open the sample data, so you will see this first table. So first table is about the first part of the lab report, in which you have to draw the ionized and acidic solution, basic solution structures of glycine, alanine, and serine. So you can use this paper to draw the structures and upload the image. The data needed for the chromatography is given here. So the 6 amino acids are given here, and the color of chromatogram is also provided here. It's either purple, dark purple, or light purple, and the distance traveled by the solvent is 3 to 3.5 centimeter. So pick one, whichever number within this range you like, you can pick that number. And then the sample distance is also given here. So this is the distance traveled by the spots or the sample. So these are the number. So what you have to do is you have to use these values. These distances are in centimeter, and you have to draw that spotted diagram in which you show that phenylalanine traveled 1.5 centimeter, and alanine traveled 1.5 centimeter, and then phenylalanine traveled 2.1 centimeter. So you have to create that picture in which the spots traveled, the distance is given here, and then that diagram or picture, whatever you're gonna create, has to be uploaded in the lab report. The data is given for 3 different unknowns. You have to use only one unknown. So whichever unknown is given to you, so please use that data. And let's go back to the lab report. So upload the image of that diagram or the graph in which you're gonna create different spots, and you're gonna show how much they moved from the bottom line. So that image has to be uploaded here. Here you have to note down the color. Again, the color of these amino acids are given in the sample data. So fill this table up, and then finally you have to calculate the RF value. As we learned, the RF value is calculated by dividing the distance traveled by the sample over the distance traveled by solvent, and you calculate the RF value for all these 6 amino acids. The distance values are given in the sample data, and you also calculate the RF value for unknown. This unknown RF value will match with one of the 6 amino acids. Whichever number it matches up with, that is the identity of our unknown. And then one of the whichever unknown is given to you, just select that, and then you have to find what amino acid is present in the unknown. So once you find that, so write down your feedback or conclusion, whatever you learned in this experiment or in this lab, and then hit submit. If you have any question or concern, please send me an email or see me during office hours. Otherwise, have a wonderful rest of the day. Bye-bye.